Okay, so this video is for AP Bio, topic 2.8 on basically osmosis and osmoregulation. So one thing that's important to think about is that all cells are surrounded in an aqueous or watery environment, and those environments vary and change throughout the course of the day or of time. And so this PowerPoint, this video is going to be about the consequences of varying external environments and solutions on cells, and also how cells can osmoregulate, or how organisms can osmoregulate or control a water balance within their cells. So let's go ahead and think about this. So here, for simplicity, we're going to talk about anything that is not water. We're going to call it a solute. So in this diagram on the left, we would talk about the sodium, the chloride, maybe the proteins, glucose. Those are all going to be solutes in the external environment of this red blood cell. Now, inside the red blood cell, there's also solutes, right? Like inside the cell, there's organelles, there's proteins, there's ions, there's going to be things that are also not water. And so when we think about molecules that we just learned about earlier in Unit 2, molecules are constantly moving, and they generally are going to have a net movement of going from a high concentration to a low concentration. And that's through like a semi-permeable lipid bilayer. So we want to remember that all cells are surrounded by these lipid bilayers, and they are what regulate what can enter and leave a cell. Now this PowerPoint is going to this video is going to focus on water moving through these lipid bilayers, kind of in response to solutes. So let's go ahead and talk about this. So generally, generally polar molecules cannot cross through the lipid bilayer because of those fatty acid tails. However, when we really think about cells are surrounded in water, and their cytoplasm is also mainly water. And because the membrane is fluid and it's flexible and it kind of like is always like moving, there's going to be a few water molecules that are able to like squeak through that fatty acid those fatty acid tails, that center region of that lipid bilayer. So some small water molecules can diffuse through the lipid bilayer directly. However, cell membranes have special protein channels called aquaporins for the more like rapid and like movement, or I don't want to say bulk transport, but for like lots of water molecules to move through that membrane at a quicker rate. So here we have these aquaporins, and this is where water molecules can really enter or leave a cell through. So water has two ways to enter or leave a cell. Very few will actually make it through the lipid bilayer, but the majority will be moving through aquaporins. Okay, so let's go ahead though and talk about some external environments in cells. So there's really three different types of external or outside the cell environments that cells may find themselves in. So the first one we're going to talk about is isotonic. So if I look at this cell here, I can see that the water concentration is pretty much equal both outside and inside of the cell. But we don't have just water, we also have ions. And solutes. And so here, the solute concentration is also equal outside and inside of the cell. But because molecules are constantly moving, the water doesn't just stay where it is. It actually is going to move into and out of the cell through aquaporins. But it really is going to be moving into and out of the cell at equal rates. It's kind of an equilibrium. We saw earlier in Unit 2 how molecules move from a high concentration to a low concentration, but here if they're equal, they're just moving in equal rates into and out of the cell. So there is no net movement of water. So our blood cells are isotonic to the plasma or the surrounding fluid in our blood. So we have equal, or our kidneys actually work really hard to keep our blood cells isotonic to their environments. And so here Water would move in and out and our blood cells would be nice and happy. This is why in like hospitals, for example, if they give patients who are dehydrated or something, they'll give them a saline solution that is isotonic to blood cells. We also may see it in like a beaker situation. So here, if you have a cell that is 2% solutes and the external environment is 2% solutes, that means they're both 98% water, and water would be flowing in and out equally across this lipid bilayer, and there'd be no change in mass of the cell. Then sometimes you may see what's called a U-tube, and if on this U-tube, there's usually like a semi-permeable membrane along the bottom, and here, if both sides are of equal concentrations, you're not going to see any net movement one way or the other. The fluids will be equal on both sides. Now, though, let's go ahead and see what a hypotonic solution is. In a hypotonic solution, there's a higher concentration of water outside of the cell. And if there's a higher water concentration outside of the cell, that means that there's less solutes outside compared to inside. So this is where our knowledge of diffusion comes into play. Water is going to diffuse from a high concentration to a low concentration within the cell. This is osmosis. And so the water is going to move through those aquaporins from a high concentration of water to a low concentration of water. So now this doesn't mean that water can't also move out of the cell through the aquaporins. It just means there's going to be a net movement. The majority of the water molecules, you're going to have more moving into the cell than you are out of the cell. Now, this added mass, right? Like these water molecules going into the cell causes the cell to swell. So this is like a gain of mass to this cell. And we'll see here on the next slide, sometimes if the cell gains too much water, animal cells could burst. So let's go ahead and see that. So here, if you put a cell into like distilled water, for example, and it's 100% water, water is going to be moving into the cell, which causes the cell to expand and swell because it's gaining water. And so here, eventually, without a cell wall, that could cause the lipid bilayer to burst open, causing a lot of water to flow out of the cell. Now, if a cell bursts, this is called cell lysis. And we don't see this in plant cells or bacterial cells because they have cell walls for support and protection, but this definitely could happen in animal cells. So for example, if like a human on a really hot day drinks a whole lot of water trying to be hydrated, and I mean like a ton of water, they run the risk of their blood becoming hypotonic and their cells bursting. People have died from this. So you want to be careful to like monitor your solute concentration and add some salts or sugars to your water. So that's why like Gatorade or something has electrolytes in it and it's not just pure water. Now here we can see if our blood becomes hypertonic, hypotonic to our blood cells, maybe from drinking too much like pure water and not enough ions, it causes our blood cells to expand and swell. It could lead to them bursting. Now, our kidneys work very hard to prevent this from happening. So if a person is drinking a whole lot of water, they're going to urinate a lot more, and that's to remove that water from our blood to try and maintain isotonic conditions. We call this osmoregulation. So same thing if like in a beaker, you put a cell into pure water, you're going to have the cell expand. Now, in a U-shaped tube, I find this always a little trippy. Here you have a semi-permeable membrane, so the solutes cannot move from one side to the other, but the water can. On the left-hand side here, we have our hypotonic solution, and water is going to move from high concentration to low concentration. So you'll actually see water like move, and it always trips me out because it actually will move against gravity and it will diffuse into, on this case, the right side, and you'll have different levels of water in that tube. All right. So water is going to move from the high concentration to the low concentration. Now, another type of environment, our last type of environment, is called a hypertonic solution. And as you can see from my picture, I have a high concentration of water within the cell and a low concentration of water outside the cell. So we can also reference this in different ways. We can say that, let's add the solute. So here we see a high solute concentration outside and a low solute concentration inside. Now, sometimes instead of saying like high solutes, you can also phrase it as a high osmolarity. So inside the cell here would have a low osmolarity. Now, water is going to flow from low osmolarity to high osmolarity. Or water is going to flow from a high water potential to a low water potential, or from a high water concentration to a low water concentration. There's a couple different ways to phrase this movement of water. So in a hypertonic situation, though, there's a low concentration of water and a high concentration of solutes outside of the cell. So this means that water molecules are in a greater concentration within the cell. So as they are colliding into each other, they are going to diffuse out. They're going to have a net movement out of the cell through those aquaporins, which causes the cell to lose mass and shrivel and shrink. So I really like this slide. So let's just watch this again. Pay attention to the cell membrane. As the water leaves the cell loses mass and it shrivels and it shrinks. So this also can easily happen in humans if a person becomes too dehydrated and is sweating and losing water to the environment. Or they have maybe you're lost at sea and you drink salt water thinking you're going to drink water, but you're adding all of those ions into your bloodstream. You create a hypertonic environment. A hypertonic environment, which then is going to cause water to leave the blood cells, and that's going to cause them to shrivel and shrink. Now, if you saw it in a beaker, you would also see the same thing. The cell would lose mass, and it will shrivel up. And then in the U-shaped tube on the right-hand side now is our hypertonic solution. And again, water is going to move from low osmolarity to high osmolarity. And therefore, it'll move through that semi-permeable membrane to the right-hand side again. Okay, now let's go ahead and just look at 3 examples or 3 visuals of what we see in animal versus plant cells. So you can see in hypotonic solutions, animal cells may undergo cell lysis and burst, whereas plant cells, they don't burst because they have a cell wall. Plant cells, actually, this is their ideal conditions and will become turgid, it's called. And so that vacuole is pressing on the cell walls, helping them to stand upright. Now, here in the opposite situation, in a hypertonic solution, the cell wall is rigid, made out of cellulose. It's for structure in the plant cell, but the cell membrane is fluid and flexible. So as water leaves a plant cell in a hypertonic environment, the vacuole shrinks. This causes a loss of mass, and now that cell membrane pulls away from that cell wall, and the plant cell kind of collapses. So this is one reason why you can't water plants with salt water, because you're adding too many solutes to their environment. Now, this is a worldwide problem as sea levels rise and more salt ocean water makes its way onto land. It contaminates aquifers and crops with ions and solutes, and it creates lots of problems for farmers throughout the world. All right, and then I thought I had a word show up for cell lysis, but obviously the animal cells bursting is cell lysis. So again, plant cells, this is their ideal conditions to be in hypotonic environments. So maybe you might be growing some plants and you think the more fertilizer you add, the better it'll be for your plants, but you want to be careful that you don't add too many nutrients to the soil because you could be creating a hypertonic solution, which will then lead to plasmolysis. Okay. Now let's go ahead and look at just a few examples of how organisms osmoregulate. Now osmoregulate means to maintain water balance. And so here we're going to look at a single-celled organism, a unicellular